In this video, we will discuss about the structure of chloroplast. In our last video, we have discussed about the endosymbiotic theory of origin of chloroplast along with the mitochondria. That is how chloroplast has entered inside the eukaryotic cell. So today, we will discuss about what is the structure of chloroplast and how does chloroplast helps in photosynthesis. So Chloroplast is a double membrane organelle that is present inside the eukaryotic cell. Double membrane means it bears two membranes. One is called the outer membrane, and the other is called the inner membrane. Now, coming to the membranes, we have already discussed that in case of chloroplast, it was actually an bacteria. So bacteria usually have only one plasma membrane. So how chloroplast has developed the two membranes, that is the outer membrane and the inner membrane. So from our last class, we know that the chloroplast is actually a bacteria that has entered into the eukaryotic cell from outside. So what is the mechanism by which chloroplast has entered into the eukaryotic cell? Endocytosis. Now, endocytosis can be broadly classified into two types. One is the phagocytosis and another is the pinocytosis. Phagocytosis deals with uptake of solid materials like virus, bacteria, or any type of food materials. Whereas pinocytosis deals with uptake of soluble materials. We will discuss about it in details when we will be studying the cell biology, but in course of the structure of chloroplast, the chloroplast has entered into the eukaryotic cell through the process of phagocytosis, because through phagocytosis particles like virus, bacteria enters into the eukaryotic cell. Now while entering into the eukaryotic cell, what happened is that suppose this is an eukaryotic cell. Suppose this is an eukaryotic cell. This is an eukaryotic cell. And suppose this is the bacteria. This is the bacteria that is willing to enter into the eukaryotic cell by the process of phagocytosis. So what will happen? The eukaryotics in the plasma membrane of the eukaryotic cell, invagination will take place. Within the plasma membrane of eukaryotic cell, what will happen? Invagination will take place, and through which the bacteria will enter into it. We will discuss the process in cell biology. I am not going to the details. And slowly this invagination will increase. Slowly this invagination will increase. This invagination will increase, and the bacterial cell will also get entered into this invagination. And finally, what will happen is that finally the bacteria will enter into the eukaryotic cell, and while entering the outer membrane of the eukaryotic cell will cover the bacteria or will enclose the bacteria, which is why the bacteria will develop the two membranes. One is the outer membrane, another is the inner membrane. So actually this bacteria is a chloroplast. So actually the outer membrane of the chloroplast is nothing but the outer membrane of the eukaryotic cell through which chloroplast has entered into the eukaryotic cell. And the inner membrane is actually the bacterial plasma membrane. So, and in the outer membrane, we know that the outer membrane does not act as permeability barrier. That is, the outer membrane of chloroplast as well as mitochondria. When we will be studying about the mitochondria in the cell biology, we will also see that the outer membrane of mitochondria or the outer membrane of the chloroplast, because both has entered into the eukaryotic cell following the same mechanism, they do not act as permeability barrier. Means any particle, if it is willing to cross the outer membrane of chloroplast or mitochondria, can easily cross the membrane. Why? Because in the outer membrane there are present certain barrel-shaped pores. There are certain barrel-shaped pore called the beta barrels, through which any molecule can enter into it. So the outer membrane do not act as permeability barrier. However, the function of the permeability barrier is performed by the actual membrane, that is the inner membrane. So we will study that all the membrane oriented functions of chloroplast are being performed by the inner membrane. The outer membrane do not perform any functions, or it does not also act as any sort of permeability barrier. Now, coming to the structure of chloroplast, this is called porine protein, which is also called beta barrel, because it is made up of beta sheets. When we study about the structure, primary, secondary structure of the amino acids, in the secondary structure there are alpha helix and beta sheet. The beta sheet participate in the formation of these porine proteins, through which large size molecules or small size molecule of any dimension can easily enter or easily cross the outer membrane of chloroplast. Now, coming to the structure of chloroplast, the basic structure or the juvenile structure of chloroplast is called proplastid. Now, what is the structure of the proplastid? Proplastid look like something like this. प्रोप्लास्टिक देखने में बहुत अच्छा तरह से अगर हम लोग ऑब्जर्व करें तो प्रोप्लास्टिक देखने में माइटोकॉन्ड्रिया जैसा ही होता है. So the structure of proplastid is somewhat like the structure of mitochondria. This is called proplastid. This is the outer membrane, this is the inner membrane, and these are the crevices which we called in case of mitochondria as cisternae. So these are the invagination of the inner membrane, invagination of the inner membrane, which in case of mitochondria we term as cisternae. So this is called proplastid, which looks like mitochondria. Now this proplastid on maturation will get converted into chloroplast. And chloroplast will have two membranes. So it also having two membranes, but the mature chloroplast does not have the foldings of the inner membrane. The invaginations of the inner membrane are absent in the mature chloroplast. Then what happens to these invaginations? When transition takes place from proplastid to chloroplast, what happened to these invaginations? These invaginations, splicing of these invaginations takes place. These invaginations are being cut down by the enzymatic activity, and they entered into the matrix of the lumen. So the invaginations are being cut down. Suppose this is the invagination and it has been cut down. And due to the hydrophobic activity, the ends are being joined up. So these invaginations on maturity are cut down and they entered into the lumen. And by the hydrophobic activity they joined down to form small vesicles inside the lumen. These small vesicles are called thylakoid. Now we know that if we consider and if we compare chloroplast in terms of mitochondria, now we know that inside mitochondria there occurs an event called electron transport chain, ETS, electron transport chain. And where electron transport chain takes place? Electron transport chain takes place in the inner membrane of mitochondria. That is, within these invaginations, electron transport chain takes place. That is, electron flows. This is called electron transport chain takes place. Similarly, within the chloroplast also, electron transport chain occurs. Means the transport of electron through series of proteins takes place, which is actually the jet scheme of photosynthesis or the light reaction. In light reaction, when we are studying the light reaction, we will be observing that in light reaction nothing but flow of electron takes place. So in mitochondria also flow of electron takes place, and in chloroplast also flow of electron takes place. The event of flow of electron in case of mitochondria is called electron transport chain, and the event of flow of electron in the chloroplast is called the light reaction of photosynthesis or the jet scheme. Now in mitochondria, the electron transport chain occurs within the cristae, within this invaginations, the inner membrane, the foldings of the inner membrane. But these foldings are absent in case of mature chloroplast. Instead of that, these foldings has been sliced out into the lumen and they have formed the thylakoids. So the event of electron transport chain in case of chloroplast will be taking place inside the thylakoid. So the event of electron transport chain will take place inside the thylakoid because the cristae are ultimately the thylakoids. There is no difference between the cristae, the foldings of the inner membrane, because in proplastis these are the cristae and which has ultimately developed into thylakoid in mature chloroplast. So in the event of electron transport chain takes place within the cristae and ultimately this cristae has developed into the thylakoid. So the event of electron transport chain in chloroplast will be taking place inside the thylakoid. Now these thylakoids, now coming to the actual structure, these thylakoids pile upon one another. These thylakoids piles upon one another and is called, this piled thylakoids is called grana. This piled thylakoid is called grana. Okay, that means the individual unit is called thylakoid and when more than one thylakoid piled upon one another, the entire structure is called grana. Sometimes two granule stack. Suppose this is a granule stack and this is another granule stack. These two granule stack may get connected with each other. This connected granule stack is termed as stroma lamellae. And the remaining lumen, the remaining lumen is called the stroma. Now in our last class we have discussed that there are two parts or the two divisions of photosynthesis. One is the light reaction and another is the dark reaction. So the event of light reaction and the event of dark reaction both takes place inside the chloroplast but at different locations. Light reaction takes place inside the thylakoid. Inside the thylakoid, light reaction of photosynthesis takes place. And within the stroma, the dark reaction of photosynthesis takes place. That is the C3 cycle. So the dark reaction takes place in the stroma and the light reaction takes place in the thylakoid. Now another thing that is to be remembered is that in the last class we have stated that the dark reaction is dependent upon the light reaction. So here the light reaction within the thylakoid will be taking place from where the thylakoid will be supplying the NADPH to the stroma, and where the dark reaction will occur. Okay, beside this, for activating the enzymes of the dark reaction, the pH of the stroma should remain in the alkaline range, that is pH 8. But generally the pH of the stroma remains in the acidic range, that is pH 5. Means under normal condition the pH of the stroma is acidic, that is pH 5. Now when photosynthesis is to be conducted, more specifically when the dark reaction of the photosynthesis is to be conducted, the pH of the stroma should get shifted from pH 5 to pH 8. And for shifting the pH, the concentration of hydrogen inside the stroma should be reduced, which is done during the light reaction. We will study in the later phase that during light reaction, when jet stream runs, an intermediate molecule called plastoquinone accept this hydrogen from the stroma and make it entered into the thylakoid. As a result of which the pH of the stroma gets shifted from acidic range to the alkaline range, that is pH 8. And under this condition, the activity of the enzymes of the dark reactions takes place. So what we have studied earlier, that the dark reaction is dependent upon the light reaction not only for NADPH but also for changing the pH of the stroma. As we have started earlier that the light reaction is light dependent reaction, so it will be taking place in the daytime. So obviously the dark reaction will also take place during the daytime, because in daytime it is only possible where the shifting of the protons from the stroma to the thylakoid could have taken place. At night, when photosynthesis does not take place, at that time this hydrogen ion from thylakoid again comes out to the stroma, and the pH of the stroma from 8 gets shifted to pH 5. We know that in case of mitochondria, the electron transport chain is associated with formation of ATP, because the complex 5 of electron transport chain is called as F0F1 particle, through which production of ATP takes place when 3 hydrogen molecules enters from the F0F1 particles. ATP synthesis event also takes place in the chloroplast also, because this hydrogen ion, when enters into thylakoid, a pool of hydrogen ion get accumulated inside the thylakoid. These hydrogen ions is liberated out to the stroma through F0 F1 particles, through F0 F1 particles, F0 F1 particles present in the thylakoid membrane. And when hydrogen ion comes out through the F0 F1 particle, it leads to the synthesis of ATP. It leads to the synthesis of ATP. So suppose this is a chloroplast having the outer and inner membrane, and the thylakoids are piled upon one another to form the grana. Beside this, the chloroplast, as it is prokaryotic in nature, will have ribosome which will be of 70S type. We know the ribosome has two subunits. One is the smaller subunit and another is the larger subunit. So, and the summation of these two subunits will be of 70S type, as it is of prokaryotic in nature. So the smaller subunit will be of 30S subunit and the larger subunit will be of 50S subunit, and the summation of which is 70S subunit. So it is prokaryotic in nature. As it is prokaryotic in nature, so the chromosome or the DNA is circular in nature. The chromosome or DNA is circular in nature, and it is called as double-stranded covalently closed circular DNA. It is called double-stranded covalently closed circular DNA. As it is circular in nature, as chloroplast DNA or cpDNA is circular in nature, so it does not follow Mendelian genetics. Does not follow the rules and principles of Mendelian genetics. This circular DNA is also resistant to an antibiotic called streptomycin. So it is streptomycin. It bears streptomycin resistance gene, by virtue of which it could protect itself from the harmful effect of the antibiotic streptomycin. Now we will study about the ultrastructure of thylakoid. Suppose this is a thylakoid, and the thylakoid has piled upon one another to form the grana. So the entire structure is called grana. Now, if we consider only a single thylakoid, and perform cross section of this thylakoid, what we will observe? That through the cross-sectional study, we will observe that the thylakoid have two membranes. The thylakoid have two membranes. One is the outer membrane and another is the inner membrane. So similar to chloroplast, the thylakoid also have two membrane, the outer membrane and inner membrane, and it will have the inner lumen, the lumen. Now on close observation of the inner membrane, means if we closely observe the inner membrane, we will observe small ball-shaped structures present throughout the inner membrane of the thylakoid. These ball-shaped structures are called quantasome. These are also called as photosynthetic units, or they are also called as light harvesting complex. That means quantasome, photosynthetic unit and light harvesting complex are nothing but the alternative names given to a same structure. Quantasome, photosynthetic unit और लाइट हार्वेस्टिंग कॉम्प्लेक्स एक ही चीज का तीन नाम है. इसका नाम क्या दिया हुआ है? फोटोसिंथेटिक यूनिट. Why? Because this is the region within the thylakoid where the light reaction of photosynthesis takes place. Now if we study a single quantasome, then what will we observe? Within a single quantasome there are three distinct layers of pigments. In the outermost layer, the molecules which are present are called carotenoids. We also consider them as antenna molecule because they are the molecule that helps to trap the solar radiation. So in the outermost layer there lies the carotenoids. Within a single quantasome, approximately 50 molecules of carotenoids remains present. In the middle layer, the molecules that are present are called chlorophyll B. Approximately 70 molecules of chlorophyll B remains present in the middle layer of quantasome, and at the middle there lies the molecules of chlorophyll A. Approximately 160 molecules of chlorophyll A resides at the center of a single quantasome or photosynthetic unit. So, the quantasome or photosynthetic unit consists of three distinct layers of molecules. In the outer layer, there lies the carotenoids. In the middle layer, there lies the chlorophyll B. And at the center, there lies chlorophyll A. And finally at the middle, there lies a special type of chlorophyll A molecule that either absorb, I am again repeating, all these molecules are chlorophyll A molecule, but at the pinpoint, at the ultimate center, there lies a special type of chlorophyll A molecule that can either absorb wavelength of light of 680 nanometer, or it can absorb wavelength of light of 700 nanometer. If the center chlorophyll A molecule is absorbing wavelength of light of 700 nanometer, then we consider them as PS1, that is photosystem 1. And if it is absorbing wavelength of light of 680 nanometer, then we consider them as photosystem 2. So a single quantasome can be either PS1 or it can be PS2. PS1 and PS2 do not lies in a single chromosome. Do not lies in a single quantasome. Quantasome for PS1 is different and quantasome for PS2 is different. So a quantasome will be of PS1 type or PS2 type depends upon the center chlorophyll A molecule, that is at what wavelength of light it is getting excited. So the carotenoids, which is present as the outermost layer in quantasome, carries out two types of pigment, carotene and another is the xanthophyll. So the carotenoids consist of carotene and xanthophyll. In the middle layer there lies the chlorophyll B, and in the innermost there lies the chlorophyll A. So in total there is approximately 280 molecules of pigments that harvest the light ultimately to either PS1 or to PS2, leading to the light reaction of photosynthesis through the conduction of jet stream. Now this quantasome bears PS1 and PS2. So now we have to study the localization of PS1 and PS2. Localization of PS1 and PS2. So from the above study, it became very clear that within the chloroplast, there lies the thylakoids, and the thylakoid piled upon one another to form the grana. And the lumen of the chloroplast is called the stroma, where the dark reaction of photosynthesis takes place. And these are the thylakoids, and the piled thylakoids are together considered as grana. Now, if we observe this surface of the thylakoid or these surfaces, closely look to the red marks that I am putting in these surfaces of the thylakoids, the sides, the lateral sides of the thylakoid, and the bottom side of the lowermost thylakoid and the top side of the uppermost thylakoid. These sides are called the non-appressed membrane. Non-appressed means, what does appressed means? Appressed means when one membrane touches the other membrane. When one membrane touches the other membrane, that is this junction site. This junction site where the membrane of the upper one thylakoid touches the membrane of the lower thylakoid, this membrane is called the appressed. But the sides that I have demarcated with the red mark are not touching anything. So they are called the appressed, non-appressed membrane, or they are considered as stromal thylakoid, or they are considered as stromal thylakoid. They are called stromal thylakoid. But these sides where one thylakoid is touching the other, these sides are called as, they are called as appressed membrane, or they are also termed as granule thylakoid. Remember this concept, because this concept will ultimately help you to understand the C4 pathway. So, in the non-appressed membrane, that is the membrane that is not touching anything, or the stromal thylakoid, here in the non-appressed membrane, the quantasomes those are present, the quantasomes that are present in the non-appressed membrane, that means these quantasomes, bears such chlorophyll A molecule at the center that can absorb light equivalent to 700 nanometer. That is, the chlorophyll A molecule get excited on receiving, or the chlorophyll A molecule getting activated on receiving wavelength of light equivalent to 700 nanometer. So it signifies PS1. So in the non-appressed membrane or in the stromal thylakoid, the quantasome bears PS1. that is photosystem 1. The quantasome participates in photosystem 1 mediated reactions. While in the appressed membrane, the quantasomes that are present in the appressed membrane can be of either PS1 type or can be of either PS2 type. That is, in the appressed membrane, both PS1 and PS2 can remain present. That some of the quantasomes acting as PS1 and some other acting as PS2. But in the non-appressed membrane, only PS1 remains present. And this evolutionary criteria ultimately led to the evolvement of C4 pathway of photosynthesis that we will study in the later half of the series. So friends, kindly remember that in the non-appressed membrane, the quantasomes bears only PS1, whereas in the appressed membrane, it may be either PS1 or it may be PS2. In the next class, we shall start a new topic. Thank you.